Synthetic biology is the construction of artificial biological pathways, organisms, or the redesign of existing natural biological systems. This begins with synthesizing artificial base pairs. All organisms are defined by their genomes, which are coded by alphabetic pairs AT and CG. Scientists have been able to expand these pairs in vitro beyond AT and CG, called unnatural base pairs, or UBPs for short. In order for this to work, the new bases must be available inside the cell, usable by the cell for DNA replication, and stable. In order to replicate artificial DNA base pairs, like natural DNA base pairs AT and CG, they must already be present in the cell. Scientists attempted to do this in two ways. The first was simple diffusion, which resulted in overexpression and poor growth. The second attempted to use nucleotide triphosphate transporters. These samples were efficiently transported into the cell, but needed to be stabilized first. Originally, the bases were decomposing in culture mediums by enzymes that use water to break phosphoric acids. Potassium phosphate was introduced and proved to stabilize the UBPs outside the cell. Next, transporters were introduced, which slowed growth but maximized uptake. This allowed concentrations inside and outside the cell to be maintained despite interior decomposition. Researchers were able to successfully replicate DNA using UBPs in vitro by engineering a plasmid to imitate specific polymerases. The engineered plasmids replaced AT pairs and positioned UBPs downstream from the origin of replication, where leading strand and lagging strand replication would be mediated by the polymerase. The UBP proved to be stable during exponential growth and stationary growth. Additionally, UBP presence did not result in reduced growth. Synthetic organisms have many significant applications. Researchers hope that the success of synthetic organisms can be a launch point for re-engineering entire cells, allow for specific nucleic acid labeling in living cells, direct the construction of tangential transcription networks, and lastly, to promote the production and evolution of new proteins.